right? So already we discussed about the pre-fertilization structure and events, and under that androecium and gynoecium, and also we saw about the pollination, that is how the male and the female gamete are brought together, and about the several agents of pollination, right? So today we'll see in detail about the, that is pollen pistil interaction. So what do you mean by pollen pistil interaction? That is how the interaction occurs between the male and the female gamete, that is fertilization, and we'll see in detail about the double fertilization and post-fertilization structure and events. So under post-fertilization structure and events, the main structures are endosperm, embryo, and other than this, the ovule will convert it into seed and ovary will form into a fruit, right? But today we'll see in detail about the endosperm and embryo. So today we are going to see about fertilization, double fertilization, then post-fertilization, particularly about the endosperm and embryo. Okay. So first, under pollen pistil interaction. So what do you mean by pollen pistil interaction? That is, the interaction exists between the pollen and the pistil, right? So it is a process in which pistil recognizes the compatible or incompatible pollen through the chemical components produced by them. So what do you mean by this? Compatible or incompatible pollen, right? So first, what happens? We saw the production of male gamete, right? How the male gamete is produced. From the pollen, that is the generative cell, it forms the male gamete. And we also saw about the production of female gamete, right? So how the female gamete is produced. In the embryo sac, that is the 8 nucleated and the 7-celled structure. So we saw the production of female gamete, right? So here in the case of pollen pistil interaction, so first in the stigma, that is the female reproductive part, the pollen comes and lands. So this is the pollen, it lands on the stigma. So this is the stigma, which is the female reproductive part, right? So it is the process in which the pistil recognizes whether the pollen is compatible or not. Because when the pollen lands on the stigma, that pollen may be either of the same species or of the different species. That is, it may be either compatible or not compatible. So that is recognized by the pistil. So when the pollen sticks to the stigma, so here the stigma secretes some substances. So the pollen swells up, right? So if the pollen is compatible, so if the right type of pollen, it lands on the stigma, the pistil accepts it. So it will accept it and it promotes the post-pollination events. So how the post-pollination events happening? That is, when the right pollen sits on the stigma, it leads to the formation of a pollen tube, right? So if the pollen tube is formed, it shows that the pollen is compatible. Okay, so if the pollen is compatible, the pistil accepts it and it promotes the post-pollination events. So the pollen grain germinates on stigma to produce a pollen tube. So how the pollen tube is produced? Through one of the germ pores. We saw the structure of the pollen, right? It is covered by an exine and internally it is an intine. So here in the exine, there will be germ pores. Germ pores will be present. So how the pollen tube is formed now? That is, there occurs a formation of the pollen tube through one of the germ pores. The pollen tube comes out of this exine through the germ pore, and the contents of the pollen grain move into the pollen tube. So here, when the pollen sits on the stigma, when it is compatible, it leads to the formation of the pollen tube. So how the pollen tube is formed? That is, it is formed through the germ pores and it moves the contents of the pollen grain. It moves into the pollen tube. Okay. So now the pollen tube grows through the tissues of the stigma. So here tissues of the stigma and style to reach the ovary. So this red line, it shows the formation of a pollen tube. So there are two types based on the number of pollen tube that is formed. That is monosiphonous and polysiphonous. So this monosiphonous means example is capsular. That is only one pollen tube is formed, example. And for polysiphonous, the example is Malvaceae and Cucurbitaceae. So only one pollen tube emerges, then the condition is monosiphonous, example capsula, and polysiphonous, that is Malvaceae and Cucurbitaceae. Okay. So based on the number of the pollen tube that is formed, it is called monosiphonous or the polysiphonous. So clear? So if the pollen is incompatible, so what will happen? If it is a wrong type of pollen, what will happen? The pistil rejects the pollen by preventing the pollen germination or the pollen tube growth. So only if the right pollen sits or lands on the stigma, there will be formation of the pollen tube. But if the wrong type of pollen it sits, then the pistil, it rejects the pollen by preventing the pollen germination or pollen tube growth. So there will be no pollen tube growth. Okay. So in plants which shed pollen grains at two-celled condition, we saw that in case of 60% of the angiosperms, what will happen? They shed at the two-celled stage. That is, the pollen grains are shed at the two-cell stage. So what are the two cells? One is a vegetative cell and another one is a generative cell, right? We saw in that the pollen grain. So if it is at the two-cell stage, that is a vegetative cell and a generative cell, the generative cells divides into two male gametes during pollen tube growth. So if the pollen is only having two cells, so now the division happens when there is a growth of the pollen tube, right? So the generative cell now divides into two male gametes. So this is the polar nuclei, and here it shows the egg apparatus and the synergids. So this is the filiform apparatus. So now what happens? The pollen tube, it enters into the this embryo sac through the synergids, right? So and it releases the male gametes. So here the fusion happens between the male gamete and the egg nucleus, and here between the male gamete and the polar nucleus, right? So it comes and it reaches the egg through the synergids. But in some cases in which the plants shed pollen in the three-celled stage, we saw that in some of the angiosperms, the pollens will be in the three-celled condition. So in that case, the pollen tubes carry two male gametes from the beginning. So initially, even when the pollen is shed, there will be the male gametes. But in the case of two-celled condition, right, when the male gametes are formed, then there is a pollen growth. Okay, so that is pollen tube growth. During that time, there occurs a formation, that is one cell, it divides to form two male gametes, that is the degenerative cell. Clear, right? So the pollen tube reaches the ovary, then it enters the ovule through the micropylar region, and then it enters through one of the synergids through the filiform apparatus. So what is the role of this filiform apparatus? The filiform apparatus, which is present at the micropylar end, that it is present at the micropylar part of the synergids, it guides the entry of the pollen tube. So this is what is happening in most of the angiosperms, right? That is the pollen tube. So how the pollen tube is formed? That is when the pollen, it sits on the stigma. The pollen uses up the nutrients of the stigma and it swells up, and at that time, the in time it pushes out and it starts to grow the, it is pollen tube through the germ pore. And first the vegetative cell is pushed into the pollen tube, and then the degenerative cell. In some cases, what happens? If the pollen is already in the 3-cell stage, there will be the male gametes. But if it is in the 2-cell stage, there the division happens and the generative cell divides to form the 2 male gametes, right? So, and it enters particularly through the, mostly in the case of angiosperms, it entered through the micropylarin to the synergids. Okay, so this is what is happening. But in some cases, it may also enter through the chalasa. So based on the mode of entry of the pollen tube into the ovule. So there are several ways in which the pollen tube, it enters into the pollen tube enters into the embryo sac, right? So based on that, there are 3 types. One is porogamy, and another one is chalasogamy, and another one is mesogamy. So this is what is commonly happening in angiosperms, that is porogamy. That is the entry through micropyle. But some cases it enters through the chalasal end. So that is called the chalasogamy. And the example is, example for chalasogamy is betula. Some plants it enters through chalasa. Or in the case of mesogamy, it enters either through the integuments. It enters through integuments. Example is cucurbita. Or through funiculus. Example is pistacia. So mesogamy is the entry of this pollen tube either through integuments or through funiculus. Then it is called the mesogamy. So based on the method of entry of pollen tube into please mute yourself. No,
110 रुपए कैशबैक का मिली तो 85
please mute yourself. Chandan Kumar.
बना लेता
So based on mode of entry of pollen tube into the ovule, it is classified as porogamy, chalasogamy, or mesogamy. So if it enters through the micropyle, it is porogamy, and if the pollen tube enters through the chalasal end, then it is chalasogamy, and if it enters either through integuments or through funiculus, then it is called the mesogamy. Okay. So the plant breeder can manipulate the pollen pistil interaction. That is how the interaction happens, even in incompatible pollination. So normally in incompatible pollinations, there will be no formation of the pollen tube. So the plant breeder can manipulate this interaction to get the desired hybrid products. Okay. So these are the different methods of entry of pollen tube into the ovule. Okay, clear? So next is the artificial hybridization. So what do you mean by artificial hybridization? So it is a crop improvement program. So whatever features we want to, that is want the plant to have, we can make it, right? So that is the artificial hybridization. So it is a crop improvement program in which decide pollen grains are used for pollination. So we are using the whatever the pollen grains we want and whatever the features we want, we can make it to happen, right? So it is a crop improvement program in which desired pollen grains are used for pollination. So this is achieved by following techniques. That is by emasculation and bagging. So what do you mean by emasculation? So emasculation is the removal of anther from the bisexual flower bed. So what do you mean by bisexual flower bed? We saw about the bisexual and then monoecious, dioecious, everything we saw, right? So here what is happening is, in the case of bisexual flower, we'll have both the male and the female reproductive structures. So what is emasculation? We are removing the anthers from the bisexual flower bed of the female parent before the anther dehisces. So before the anther dehisces, the anthers are removed. So only the female part of the plant will be present, right? So here the emasculated flowers are covered with a suitable bag. So we are covering it with the bag after removing it with the forceps, and we are covering it with the butter paper or with some bag to prevent the contamination of its stigma with unwanted pollen. So if it is open, what will happen? The stigma will receive any pollen, right? To prevent it, we are covering it with a suitable bag to prevent the contamination. So then when the stigma attains the receptivity, that is when it is in a position to accept the pollen. So now the mature pollen, so whatever the characteristic features we are in need of it, we are collecting that pollen. That is what type of plant we want, right? So those pollen grains are collected. That is the mature pollen grains are collected from anthers of male parents. So they are taken from the male parent and they are dusted on the stigma. So we are removing the bag and dusted it over the stigma. Okay, so then the flowers are again rebagged. Then it is again sealed and allowed to develop the fruits, right? So now the stigma is, now it is fused with, or it is landed with the pollen of a particular character that we want, right? So that is the artificial hybridization. So now the flowers are again rebagged and allowed to develop the fruits. So this is the method of that is emasculation and bagging. So for the case of unisexual flowers, so unisexual flowers will have either the male reproductive structure or the female reproductive structure, right? So there is no need for emasculation because already it will have only the female reproductive part if it is a female flower. So female flower buds are bagged before the flowers open. So we are bagging the female flower buds because if it is open, it will receive any type of pollen. So to prevent it, the female flower buds are bagged before the flowers open. And then when the stigma, it become receptive, when it is in the ability to accept the pollen, the pollination is carried out. The pollination is carried out using the desired pollen and the flower is rebagged. So you are making the particular pollen to land on a particular stigma so that we can get the desirable characteristics. So that is called the artificial hybridization, and this is for the crop improvement program. Okay, clear? So clear with it, how the pollen pistil interaction happens and how it selects the correct type of pollen. That is, pistil can recognize the correct type of pollen, right? So after it selects the correct type of pollen, there occurs the formation of the pollen tube, right? And it reaches the ovule. So based on that, there are three ways. Either it may be a porogamy, chalosogamy, or mesogamy, and how the artificial hybridization is carried out. Clear with it? So the next is the double fertilization. So why it is called double fertilization? Because there occurs in two fertilization events, right? So how it is happening? After entering one of the synergids, so we saw that in majority of the angiosperms, so how the pollen tube enters the ovule, that is through the synergids, right? After entering one of the synergids, the pollen tube, it releases two male gametes into the cytoplasm of the synergids. So it releases its two male gametes. So how the male gametes are formed? It is formed by the division of the generative cell, right? So it releases the two male gametes into the cytoplasm of the synergid, right? So among the one male gamete, it moves towards the egg cell. So it fuses with the egg cell and fuses with its nuclei, and this process is called syngamy, and to form the zygote. And this syngamy is also called generative fertilization. This is also called generative fertilization. Okay. And the other male gamete, it moves towards the two polar nuclei. We saw the structure. I hope you're clear with the structure, right? In the middle, there will be two polar nuclei. And at one end, that is at the micropylar end, we'll have the synergids and the egg nucleus. So one male gamete, it fuses with the egg cell and to form the zygote. So it is called generative fertilization. The other male gamete, it moves towards the two polar nuclei that is present in the central cell, that is in the central large cell, and fuses with them to produce a triploid primary endosperm nucleus, which is also called PEN, that is primary endosperm nucleus. So we are calling it as triploid because the male gamete is a haploid and it fuses with the two polar nuclei. So that is N. One polar nuclei is one N and another polar nuclei is another N, another haploid, right? So to form a triploid structure, which is called the triploid primary endosperm nucleus, right? So as it involves the fusion of three haploid nuclei, it is called triple fusion. So this process is called triple fusion, and this is also called vegetative fertilization. Vegetative fertilization, right? Because here this fertilization, that is the egg with the male gamete, that leads to the formation of zygote. So it is a generative fertilization, and this is called the vegetative fertilization. Okay. So this double fertilization, actually, it was reported by the scientist, that is Navasan, in Fritillaria and Lilium, in plants, Fritillaria and Lilium. Okay, since two types of fusions, that is syngamy and triple fusion, it take place in the embryo sac. Even in the single embryo sac, two types of fertilizations are happening. So it is called double fertilization, and also it is a unique event in flowering plants, that is angiosperms. Only the angiosperms, they show this feature of double fertilization, right? So it is an important event. So it involves the two types of fusion. So it is clear, right? So how it is happening, and even some problems they may ask based on the haploid or the triploid or diploid nature of this. There is questions maybe with the calculations, with the problems like. So you have to be clear with the which one is haploid and what are the cells which are diploid and which one is triploid. Okay, so here the triploid is only the endosperm, and the central cell after triple fusion. So after this fusion, it became the primary endosperm cell. So the cell is called primary endosperm cell, and it develops into endosperm. So this is what is transformed into endosperm. So this is happening after fertilization, that is post-fertilization structure. And zygote, it develops into what? Embryo, right? And ovule, it forms the seed, and ovary, it forms the fruit. So all these are the post-fertilization events or the post-fertilization structures. So I hope you're clear with this, right, till now. So we'll proceed with post-fertilization structure and events. So what are the post-fertilization? That's what we saw now, right? Endosperm, then embryo, and then seeds, and then fruit. Right. So first we'll see in detail about the endosperm development. So how the endosperm is formed. So we saw that the primary endosperm cell, it divides repeatedly to form a triploid endosperm tissue. So it forms a diploid, that is triploid structure, right? 3N. Triploid is 3N. And endosperm cells are filled with reserve food material. So the main role of this endosperm is to provide nutrition to the growing embryo, right? So this is what is happening after fusion or fertilization. So first, here we saw that egg cell, when you see the structure of the embryo sac, right? First, initially it will be a egg cell and two synergids will be present. So after fertilization, what happened? The synergids, it starts to degenerate. So degenerating synergids, and the male gamete fuses with the egg cell and it forms a zygote. So it is transformed into a zygote now. And here what happens? It became a primary endosperm cell. Previously, it was a large central cell. Now that has become a primary endosperm cell. And this is a primary endosperm nucleus, which is also called PEN. So how this is formed? One male gamete fuses with the two polar nuclei to form a primary endosperm nucleus. And after fertilization and another changes, this antipodal cells, they also degenerate. So degenerating antipodal cells. So this is the structure of the embryo sac after fertilization. Okay. So here this endosperm cells, actually they are filled with reserve food materials. Okay. So what is the role of it? This zygote, now it develops into an embryo. For the growth of the embryo, they need the nutrition, right? So that is provided by this endosperm cells. So they provide nutrition for the developing embryo. Okay. So in common endosperm development, PEN, right? We saw the PEN. So this PEN, it undergoes successive nuclear division to give rise to free nuclei. So what do you mean by free nuclei? Only the nuclear division will happen, but there will be no cytoplasm or there will be no cell wall formation. So that is called the free nuclear endosperm. So the nucleus divides many times to form a free nuclear endosperm. So the number of free nuclei varies greatly. Then what happens? This endosperm becomes cellular. So this nuclear endosperm is converted to cellular endosperm by the wall formation. So first, inside there will be many nuclei, right? There will be many nuclei without wall. Then there occurs the formation of the wall and each one forms into a cell. So it is converted into a cellular endosperm. So the common example is the tender coconut water. So if you take the tender coconut water that you would have seen, right? So the inside water is having, actually it is having thousands of nuclei. That is, it's a free nuclear endosperm. But the surrounding white kernel is a cellular endosperm. That is, there occurs a formation of the wall. So here it is a free nuclear endosperm where thousands of nuclei are present. So based on the development of the endosperm, so there are different types, like one is called the nuclear endosperm. So this is the nuclear type, then cellular type and halobial type of endosperm. So these are the different types of endosperms that are present in angiosperm. So what is happening in nuclear endosperm? So in nuclear endosperm, so here it is a free nuclear division. So this is the micropylar end, and here is the first free nuclear division happens. That is, only the nucleus divides. So first it is a free nuclear division. So how it will be? It is a multi-nucleated structure. That is, many nuclei will be present. So it is a multi-nucleated endosperm, and then after the formation of many nuclei, it undergoes cytokinesis. Cytokinesis takes place, which leads to the formation of wall formation. So multinucleated, now it becomes a multicellular endosperm at maturity. Right? So this is one form of endosperm. So first there occurs a nuclear division. There will be no cell wall formation. Then there occurs a wall formation. So leads to the formation of cellular endosperm at maturity. And second type is cellular endosperm. So here the main difference is, this is found in gamopetalae group. This is found in cellular endosperm, is found in gamopetalae. Here the example for nuclear endosperm is capsula. And mostly dicotyledons, they show nuclear endosperm. Okay, so it is found in gametopetale group. So here what happens? After the division, that is after the formation of the nuclear division, after every nuclear division, there occurs an wall formation. So if you see, there will be a single nucleus which is surrounded by the wall, right? So that forms a separate cell. So it is found in gametopetale, and each division of primary endosperm nucleus EN is followed by cytokinesis. So it forms the cellular. Right. So even from the beginning, there occurs here the multicellular endosperm will be present only at maturity. So here from the beginning, there will be the presence of cellular endosperm. That is after the nuclear division, it is followed by cytokinesis. So that is cellular endosperm. Right. The third type is halobial. So it is a halobial endosperm. So here first, this primary endosperm nucleus division is followed by the unequal cytokinesis. First one is unequal cytokinesis. So then what happens is free nuclear division. Free nuclear division, then which is turn is followed by cytokinesis. So it is the intermediate type of endosperm which is mainly present in monocots. It's intermediary type. So here what happens? First there is an unequal cytokinesis, one larger cell and one smaller cell, which is followed by free nuclear division, that is no formation of the wall, and then cytokinesis, which leads to the cellular endosperm at the maturity. Okay, so it is intermediary type and it is found in monocots. So different types of endosperms are present, right? So one is the nuclear endosperm and another one is the cellular endosperm, and third one is the halobial endosperm. Okay, so based on the type of division, that is nuclear division or the which is followed by cytokinesis at the beginning or at the end. So based on that, there are different types of endosperms. Okay, clear? So next type of the post-fertilization structure is the embryo development. So how the embryo development occurs? The embryo develops at the micropylar end of the embryo sac, right? So we know the structure right first here. So this is the micropylar end. So the embryo starts to develop from the zygote. So this already we saw, right? After fertilization, this is a structure. That is the degenerating antipodal cells and degenerating synergids, and the egg cell, it fuses with the male gamete and it forms an zygote. So this zygote develops into one embryo. So it develops at the micropylar end of the embryo sac where the zygote is situated. Okay, so most zygotes, it divides only after the formation of some endosperm. So what is the role of it? First, there occurs in formation of the endosperm. So first polar nuclei, it fuses with the male gametes and leads to the formation of the endosperm, right? So what is the role of it? Only after the endosperm is formed, the zygote starts dividing. This is because so that they can get nutrition from the endosperm, right? So this endosperm, it gives nutrition to the developing embryo. So in monocots and dicots, if you see the seed structure, they differ greatly, right? For example, if you see the structure of the monocot and the dicot seed, there will be a great difference. But if you see the embryogeny, so what is embryogeny? That is the early development of embryo. From the zygote, it forms an embryo, right? So this early embryonic development is similar in both monocots and dicots. So what is happening? How the embryo is formed? First, the zygote, it divides into two. That is transverse division. The first division is the transverse division. division, and it forms two cells. One is called the basal cell or the suspens. So the upper one is called the basal cell or suspensor cell. And the other one, the down, that cell is called the apical cell or terminal cell or embryonal cell. So now this embryonal cell or the cell which is present at the bottom here, right? So this is the embryonal cell or apical cell or the terminal cell. So this embryonal cell, it divides mitotically. This divides mitotically to form a proembryo. So first it forms a proembryo, which then forms a globular embryo. Then it forms an heart-shaped embryo, which is converted into one mature embryo. So here what happens? That is, there occurs a mitotic division in the embryonal cell. So it leads to the formation of proembryo, globular embryo, heart-shaped embryo, and mature embryo. Okay, then this basal cell or the suspensor cell, that undergoes division about 6 to 10 cells. So you can see the structure, right? So about 6 to 10 cell structure, and it forms a filamentous structure that is called the suspensor. So this is the suspensor, and these structures, it may be from 6 to 10, and they form a filament-like structure that is called the suspensor. And what is the role of this suspensor? That is suspensor. The main function is to push the developing embryo. Is to push the developing embryo into the endosperm, into the food-laden endosperm, to provide nutrition, right? That is, the main role of this suspensor is to push the developing embryo into the endosperm, so that it can get its nutrition. So the endosperm is the one which provides nutrition for the growth of the zygote to form an embryo, right? So the suspensor, it pushed the developing embryo into the endosperm to provide nutrition. So this is what is the development of embryo, right? So first, the zygote, it undergoes transverse division to form two cells, that is basal cell or the suspensor cell, and the apical cell or terminal cell or embryonal cell. Then this embryonal cell, it divides mitotically. There are so many divisions, right? Mitotic division to form an proembryo, and then it forms a globular embryo, heart-shaped embryo, and then into mature embryo. And then this suspensor cell, actually it divides, that is undergo mitotic division 6 to 4 nucleated cell. It forms a filament-like structure, and the main role of this suspensor is to push the developing embryo into the food-laden endosperm to provide nutrition. Okay. And if you see the structure of a dicotyledonous embryo, that is, it will have the two cotyledons, right? So it has an embryonal axis and two cotyledons, and the portion of the embryonal axis which is above the level of cotyledon is called the epicotyle. So which is above is epicotyle, and that will develop into a plumule. So this is the structure of the plumule, which grows into a shoot or a stem, right? And then the cylindrical portion, which is below the cotyledon, is called the hypocotyle, and which terminates with the radicle or the root tip. So this is the radicle, and which forms the root tip, and that is covered by the root cap. So this is in the case of a dicotyledonous embryo, right? And what about the monocotyledonous embryo? So this already actually we saw under angiosperms, right? So I hope you are clear with these terms: epicotyle, hypocotyle, radicle, and plumule. So here they possesses only one cotyledon, And the cotyledon of the grass family is called scutellum. That is the outer covering is called the scutellum. So this is the scutellum which is present in grass. And it is situated lateral to the embryonal axis. And the lower end, the embryonal axis is a radicle and the root cap. But here, particularly in the case of monocots, they are enclosed in coleorhiza. So this structure is called the coleorhiza. So this is the radicle and this is the root cap, and they are covered by an undifferentiated sheath which is called coleorhiza. And the portion of the embryonal axis which is above the level of attachment is called the epicotyle, and they are enclosed in the coleoptile. So here it is coleoptile and here it is coleorhiza, right? So they are covered by a undifferentiated sheath which is called coleorhiza, and here coleoptile. So these terms are more important here. What is scutellum, what is coleorhiza, and what is a coleoptile? Okay, so clear with these. So this is about the structure of monocotyledonous and the dicotyledonous embryo. And an axis which is present between the plumule and the radicle is called the embryonal axis. Embryonal axis, which is also called tigellum. That is axis which is present between the radicle and plumule. And plumule. Okay, so this is about the structure of embryo. So any doubts till now? Right, so we'll discuss few questions. All of the following include outbreeding devices except. So all of the following include outbreeding devices except unisexuality of flowers, self-incompatibility, pollen release and stigma receptivity are not synchronized, anthers and stigma are placed at different position, then bisexuality, homogamy and cleistogamy. So all of the following include outbreeding devices except. Yes, B is the right answer. So why? Because this unisexuality of class and self-incompatibility, these are all the devices for to ensure the cross-pollination, right? So if it is an unisexual, obviously only cross-pollination can occur. Self-incompatibility, that is if it is from the same, belong to the same species, the same pollen and same stigma, there will be no formation of pollen growths. So this is an outbreeding device. Then pollen release and stigma receptivity are not synchronized, then that leads to cross-pollination, right? Anthers and stigma are placed at different positions. So this is also one of the methods. But bisexuality, homogamy is also one. Inbreeding and cleistogamy, so the flowers are fully closed, so only autogamy is possible. So all these features will lead to self-pollination, right? But these features, they lead to cross-pollination. So they are all outbreeding devices. So bisexuality, homogamy and cleistogamy is the right answer. Repeated self-pollination over the generation produces new varieties, better progenies, inbreeding depression, elimination of weak traits. So here the correct option is yes, C is the right answer. So inbreeding depression, repeated self-pollination over generation produces inbreeding depression. So it will not lead to the formation of new varieties, or there will be no better progenies if there is more self-pollination, and also there will be enhancement of weak traits and not the elimination of the weak traits. So correct answer is inbreeding depression. Which of the following option is correct? Right? Pollination gives a guarantee of the promotion of the post-pollination events that lead to fertilization. The events from pollen deposition on stigma until the pollen tubes enter the ovule are together referred to as pollen-pistil interaction. Pollen-pistil interaction is a dynamic process involving pollen recognition followed by only promotion of the pollen and not the rejection. Pistil has no ability to recognize the pollen, whether it is right or wrong. So, which is the right option here? Yes, B is the right option. So, B is the right answer. Because the events from pollen deposition on the stigma until the pollen tube enter the ovule are referred to as pollen-pistil interaction is the right answer. So if you take the option A, pollination gives the guarantee of the promotion of post-pollination events that lead to fertilization. So actually pollination will not guarantee, right? So it must be a right type of pollen, then only the post-pollination events will lead to fertilization. So that is the incorrect statement. Pollen-pistil interaction is a dynamic process involving pollen recognition followed by not only promotion. So if it is a wrong pollen, that will lead to the rejection also. So it is also an incorrect statement. And pistil actually has the ability to recognize the pollen, whether right or the wrong. So this is also a wrong statement. So the correct statement is option B. Okay. The total number of nuclei involved in double fertilization is, so how many nuclei are actually involved in double fertilization, whether it is 2, 3, 4 or 5. How many nuclei are involved in double fertilization? So you think of the process, how the fertilization is happening. So what is double fertilization? The nuclei, that is one male gamete, it fuses with the egg nuclei to form a embryo, right? The another male gamete, it fuses with the two polar nuclei. No, it is not option B. It is actually option D. It is actually a general question. Like it is not clearly asked as only in the embryo sac. If you see in the embryo sac, it is 3 nuclei involved. But it is generally asked the total number of nuclei. So that is 1 nuclei from the male gamete, right? 1 from male gamete and 1 from egg, which leads to the formation of zygote. Then 1 male gamete, 1 male gamete plus 2 polar nuclei. So this leads to the formation of the primary endosperm nucleus. So here 1 nuclei and here 1 nuclei, 2 nuclei and here 1 nuclei and 2. So totally 5 nuclei are involved. So D is the right option. So are you clear? So there are, that is 3 nuclei from the embryo sac, right? And 2 nuclei from the male gametes. So totally 5 nuclei are involved in double fertilization. So D is the right answer. Okay. So if you see the question and then answer, if it is asked as how many nuclei in the embryo sac is involved, then your answer is right. That is only 3 nuclei are involved. But if it is a general question, then you have to include the number of nuclei present in the male gametes also. Okay. So based on the question, you have to answer. Yes, next question is double fertilization is unique in pteridophytes, bryophytes, gymnosperms, and angiosperms. So double fertilization is a unique event in D, that is in angiosperms of the flowering plants. So D is the right answer. In coconut liquid, nuclear endosperm is surrounded by white kernel, which is integument or seed coat, cellular endosperm, lobial endosperm, fibrous mesocarp. So in coconut liquid, nuclear endosperm is surrounded by white kernel, which is an integument of the seed coat, cellular endosperm, lobial endosperm, and fibrous mesocarp. Yeah, it is a cellular endosperm. First, there will be a nuclear endosperm, which is a liquid part is a nuclear endosperm, and that is surrounded by a cellular endosperm. So D is the right answer. Okay. The study of formation, growth, and the development of new individuals from the egg is called embryology, embryogenesis, morphogenesis, and embryolysis. The study of formation, growth, and development of new individual from egg. Yes, it is A. It is embryology. So the study is called embryology. Embryogenesis is the formation, right? So study of formation, growth, and development of new individual is called embryology. Then the pollen tube enters the embryo sac usually by penetrating the egg cell through one degenerated synergid by destroying the antipodal cells between persistent synergid and central cell. So how the pollen tube generally it enters the embryo sac? Yes, B is the right answer. That is, first there occurs a degeneration of the synergid, and through that the pollen tube enters into the embryo. Right? So clear? So any doubts in this? I will see a few more questions. Okay. So the next question, the chasmogamy refers to the condition where the flowers remain closed, flowers absent, flowers open, flowers are gamopetalous. Chasmogamy refers to the condition where option A, flowers remain closed, flowers absent, flowers open, flowers gamopetalous. So it is the correct option in the case of chasmogamy. Chasmogamy. B. No, the flowers remain closed. Flowers, yeah, flowers open. The option is option C actually. Flowers are open, right? So the flowers will be open. Yes, when pollen grains of a flower are transferred to stigma of another flower of a different plant, the process is called zytonogamy, xenogamy, autogamy, homogamy. Yes, xenogamy is the right answer. When it is transferred to the stigma of another flower of a different plant, so it is xenogamy. Maize is the best example of anemophily, ornithophily, entomophily, hydrophily. Maize is a best example of it is an anemophily, and the longest pollen tube also is formed in maize. Maize is an example for anemophily. Which of the following promotes the pollen germination and tube growth? Starch, boron, calcium, and potassium. Which of the following promotes the pollen germination and tube growth? Yeah, it is boron. Boron is actually involved in the pollen germination and tube growth. Pollen germination and the tube growth. So boron. And the optimum temperature for the formation of this pollen tube is 20 to 30 degrees centigrade. Okay, so boron is helpful, or it promotes the pollen germination. The main embryo is developed as a result of pollination, triple fusion, syngamy, fusion of two polar nuclei of an embryo sac. So the main embryo is developed as a result of pollination, triple fusion, syngamy, fusion of two polar nuclei of an embryo sac. So which is the right option? Is C, syngamy. The main embryo is developed as a result of syngamy, right? Then process of double fertilization was discovered by Holmeister, Strasburger, Navashin, and Anite. The process of double fertilization was discovered by A.S. Navashin is the right answer. Option C. After fertilization, the seed is developed from the ovule, ovary, nucellus, and endosperm. After fertilization, the seed is developed from ovule, ovary, nucellus, and endosperm. Yeah, A is the right answer. That is ovule. Double fertilization means fusion of male gamete and ovum, fusion of two polar bodies, and a male gamete fused with egg and second male gamete fused with secondary nucleus, and all the above. So which is the right option? The double fertilization means, yeah, C is the right answer. That is the double fertilization means a male gamete fused with egg and the second male gamete fused with the secondary nucleus, right? C is the right answer. The fusion product of secondary nucleus and the male gamete is nucellus, primary endosperm nucleus, zygote, and secondary nucleus. Yes, PEN or the primary endosperm nucleus is the right answer. Water of coconut is endosperm, nucellus, endocarp, mesocarp. Water of coconut is, yeah, it is A, which is endosperm. The suspensor in capsula develops from apical cell, basal cell, chalosyl cell, apical and basal cell both. Yes, it is from basal cell. Basal cells which leads to the formation of suspensor, right? The number of nuclei taking part in double fertilization. So this question actually is all right. 2, 3, 4, 5. The number of nuclei taking part in double fertilization is, so just now we saw it is 5. Yes. Then, the endosperm of angiosperm is 2n, 3n, n, 4n. The endosperm of angiosperm is 2n, 3n, n, and 4n. Yeah, B, that is 3n. The entry of pollen tube through micropyle is called porogamy, syngamy, chalosogamy, and mesogamy. So, which is the right option? The entry of pollen tube through micropyle is called porogamy is the right answer. The arrangement of the nuclei in a normal embryo sac in the dicot plants is option A, 2 plus 4 plus 2, 3 plus 2 plus 3, or 2 plus 3 plus 3, or 3 plus 3 plus 2. So, which is the right option? The arrangement is, that is a arrangement of nuclei in a normal embryo sac in a dicot plant is. So, the type of arrangement is actually 3 plus 2 plus 3. So, option B. Yes, you have to be clear with it because some options are given as 2 plus 3 plus 3, or it is given as 3 plus 3 plus 2, and one more option is 2 plus 4 plus 2. So the correct option is here because at one terminal there will be 3 and in the center there will be 2, right? And at the another terminal it will be 3. So this is the correct answer. So one is the egg apparatus, the other one is the antipodal cells, and in the middle there will be 2 polar nuclei, right? So 3 plus 2 plus 3. In a cereal grain, the single cotyledon of embryo is represented by coleorhiza, scutellum, profil, and coleoptile. In a cereal grain, yeah, B is the right answer. That is scutellum. The male gametes in angiosperms are formed by the division of microspore mother cell, microspore generative cell, and vegetative cell. So, which is the right option? The male gametes in angiosperms are formed by the division of, so how the male gametes are formed? Either by the division of microspore mother cell, microspore generative cell, and vegetative cell. So the correct option is actually option C, which is a generative cell. Sir, generative cell. So the question is, yeah, C is the right answer. From microspore mother cell actually first what forms? They lead to formation of the microspore, then pollen grain, and in the pollen grain then again it divides to form vegetative cell and the generative cell. So this generative cell it divides to form 2 male gametes, right? So generative cell is the, that is it divides to 2 male gametes. So C is the right answer. Okay. So which of the following is surrounded by a callus wall? Pollen grain, microspore, mother cell, male gamete, and egg. Which of the following is surrounded by a callus wall? Pollen grain, microspore, mother cell, male gamete, and egg. So callus wall is actually B, that is microspore mother cell. So where actually callus is present? We saw, right, first the microspores, they form a tetrad structure. So those tetrads that are surrounded by the callus, and they are now split to form the individual microspores, that is tetrad to individual microspores, and is dissolved by the callase enzyme, which is produced by the tapetum, right? So actually it is not the pollen grain. Pollen grain has the exine and intine layer and not the callus wall. Okay, so the answer is B, that is microspore mother cell is the right answer. Clear? So any doubts in this? Clear with this, right? And also you be clear with the haploid, diploid, and the triploid also. The only the 3N structure is endosperm. So if they ask about the chromosome number, then you have to put it as the triple times, right? So it's a triploid. So in that you have to be clear here with the condition, whether it is a haploid, diploid, or a triploid. Okay, so clear with that, right? So tomorrow we'll see about the other post-fertilization structures like seed and the fruit, and also about the other features like what is apomixis, then polyembryony. So by tomorrow we'll finish up this chapter, that is sexual reproduction in flowering plants, right? So you're clear with it till now? So we saw the pre-fertilization, then fertilization, and under post-fertilization, endosperm and embryo, right? So you go through this, and if you have any doubts, maybe we can discuss tomorrow also. So clear till now? Okay then, thank you.